Hello viewers, welcome to Biochemist Biochemistry classes. Today I am back with a video tutorial on gluconeogenesis. Gluconeogenesis, as the name suggests, it means the synthesis of new glucose molecule from non-carbohydrate sources. We know that there are various organs and tissues in our body that depend on continuous supply of glucose as a metabolic fuel, like our brain, erythrocytes, exercising muscle. Now, if we are on prolonged fasting or starvation, the liver glycogen will get depleted after 10 to 18 hours, and then we require some other metabolic fuel to get converted to glucose and serve the demand of our body. During that time, gluconeogenesis is very important for our physiological system. Now, where does gluconeogenesis take place in our body? It takes place mainly in the liver and the renal cortex. In the cell, it is one of the pathway that is partly cytosolic and partly mitochondria. Along with this, there are two other pathways in our body that are partly cytosolic and partly mitochondria. These are heme synthesis and urea cycle. This is a common question in Viva table. What are the substrates for gluconeogenesis? Gluconeogenesis takes place from lactate, from glycerol, from propionyl coenzyme A, and from glucogenic amino acids. Now, let us check the steps of gluconeogenesis. Gluconeogenesis is not simple reversal of glycolysis. Why so? Because the formation of glucose does not occur by a simple reversal of glycolysis, because energetically the equilibrium of glycolysis is strongly favored. That is, it is more inclined towards the conversion of glucose to pyruvate. Instead, glucose is synthesized by a special pathway that requires the involvement of both mitochondrial as well as cytosolic enzyme. Moreover, there are three irreversible pathways of glycolysis which need to be overcome to get converted to glucose. Now, let us check what are the steps that are irreversible in glycolysis. The conversion of phosphoenol pyruvate to pyruvate by the enzyme pyruvate kinase, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase 1, and the conversion of glucose to glucose 6-phosphate by the enzyme hexokinase. These three steps are the major roadblocks in cases of glycolysis. If you want to know the details of this step, then you can go to my video tutorial on glycolysis. The link is given in the description box or in the I button above. Let us check what are the steps of gluconeogenesis. Pyruvate will get converted to oxaloacetate via carboxylation, and the enzyme required for this reaction is pyruvate carboxylase. Now, if you remember, any carboxylation reaction requires ABC. That means A for ATP. So, here also ATP will get converted to ADP, biotin, as well as carbon dioxide. Now, this oxaloacetate will again get converted or reduced to malate by the enzyme malate dehydrogenase, and here NADH will get oxidized to NAD+. Now, these steps up to the conversion of malate will take place in the mitochondria. These steps, they will take place in the mitochondria, and from the malate that is formed in the mitochondria, we need to get it again converted to phosphoenol pyruvate, but this steps cannot take place in the mitochondria, so malate will get transported to the cytosol and again it will get reoxidized to oxaloacetate. The same enzyme malate dehydrogenase will act here and NAD will get reduced to NADH. Now this oxaloacetate will be acted upon by the enzyme phosphoenol pyruvate carboxylase and it will get converted to phosphoenol pyruvate. Here GTP will get converted to GDP. Now one first irreversible step, if we consider in the glycolysis from the terminal end of the pyruvate, that has been taken care of. We have got phosphoenol pyruvate. Now rest of the steps will be reversible until and unless it is again converted to fructose 1,6 bisphosphate. Now this fructose 1,6 bisphosphate will be converted to fructose 6-phosphate by the enzyme fructose 1,6 bisphosphatase. Now this fructose 6-phosphate will get isomerized to glucose 6-phosphate. That is also an reversible step, and then again we have the third irreversible step. The glucose 6-phosphate will get converted to glucose by the enzyme glucose 6-phosphatase. Now this enzyme glucose 6-phosphatase, this is physiologically absent in muscle. So from muscle we cannot directly have the glucose, free gluconeogenesis from different substrates. From lactate, lactate is converted to pyruvate by the enzyme lactate dehydrogenase. From glycerol, glycerol is converted to glycerol 3-phosphate by the enzyme glycerol kinase. Now this glycerol 3-phosphate will get converted to dihydroxyacetone phosphate, and this dihydroxyacetone phosphate will get isomerized to glyceraldehyde 3-phosphate, and that will get converted to fructose 1,6 bisphosphate. So from glycerol ultimately we get fructose 1,6 bisphosphate, and this will again be acted upon by the fructose 1,6 bisphosphatase and glucose 6-phosphatase to form glucose. From lactate we get pyruvate, and pyruvate will involve all the steps of gluconeogenesis. From propionyl coenzyme A. Propionyl coenzyme A, if we consider it, it is an end product of the oxidation of odd chain fatty acid. Now this propionyl coenzyme A will get carboxylated to methyl malonyl coenzyme A by the enzyme Propionyl Co-A carboxylase, and that will get isomerized to succinyl coenzyme A. Now this requires the enzyme methyl malonyl coenzyme A mutase. This enzyme requires vitamin B12 for its catalysis. It requires vitamin B12 in the deoxyadenosyl form. The absence of this enzyme or sometimes the absence of vitamin B12 from diet may lead to methyl malonyl aciduria. Now this succinyl coenzyme A will get converted to oxaloacetate, the metabolic hub in the metabolic hub of TCA cycle. Now this oxaloacetate will again get converted to phosphoenol pyruvate via the malate, and phosphoenol pyruvate carboxylase enzyme will act on it. So this is how gluconeogenesis can take place from lactate, from glycerol, and from propionyl coenzyme. Glucogenesis may take place from different substrates like amino acids. Amino acids, we all know they are derived from the hydrolysis of tissue proteins and are the major sources of glucose during a fast. The amino acids may get converted to oxaloacetate or alpha ketoglutarate, and this alpha ketoglutarate, it is an intermediate of the TCA cycle, so that may again get converted to oxaloacetate, and from oxaloacetate glucose can be synthesized by the different enzymes of gluconeogenesis. Other than lysine and leucine that are purely ketogenic, rest of the amino acids have some potential to form glucose. Like glycine, serine and alanine, they can get converted to pyruvate, and from pyruvate glucose can be synthesized. Valine, isoleucine and methionine, they get converted to succinyl coenzyme A. Phenylalanine, tyrosine can get converted to fumarate, and arginine, histidine and proline, they get converted to glutamate, and from glutamate to alpha ketoglutarate. All these products of the amino acid, they are linked to the metabolic hub of Krebs cycle, and they can get converted to oxaloacetate, and from oxaloacetate glucose can be synthesized. Aspartate and asparagine can directly get converted to oxaloacetate, and from that we can jolly well synthesize glucose. So this is how from amino acids gluconeogenesis can. Now, very important part, that is regulation of gluconeogenesis. Like every metabolic pathway, gluconeogenesis may have long-term regulation as well as short-term regulation. Long-term regulation is by induction and repression of the enzyme. As we know, insulin is inhibiting the gluconeogenesis. That means it will try to taper down the glucose level in our blood. So it will repress the transcription of the messenger RNA of the phosphoenol pyruvate carboxylase enzyme, that is the rate limiting enzyme of gluconeogenesis. While the antagonistic activity, that is glucagon, will induce the mRNA of the same enzyme and increase its transcription. The short-term regulation can be done by covalent modification. Covalent modification can be done via phosphorylation and dephosphorylation. Now, glucagon via an activation of cyclic AMP dependent protein kinase, it will stimulate the conversion of the enzyme pyruvate kinase to its inactive form, and this decreases the conversion of phosphoenol pyruvate to pyruvate, which has the effect of diverting phosphoenol pyruvate to synthesis of glucose. What does it mean? That means that glucagon, that will directly, what it will do, it will phosphorylate the enzyme pyruvate kinase, and pyruvate kinase is inactive in phosphorylated form. Now, what this pyruvate kinase do? This pyruvate kinase, it causes the conversion of phosphoenol pyruvate to pyruvate. Now, the pyruvate formation will be inhibited, so there will be more and more amount of phosphoenol pyruvate in the cell, and now this phosphoenol pyruvate will enter the gluconeogenesis pathway. Allosteric activation is another type of, or allosteric regulation is another type of short-term regulation. Allosteric regulation can be done via Acetyl Co-A. Hepatic pyruvate carboxylase is allosterically activated by acetyl CoA during fasting, and it is inhibited by the fructose 2,6-bisphosphate. So these are the important steps of gluconeogenesis as well as its regulation. To sum up, gluconeogenesis take place in liver and renal cortex. It is partly cytosolic and partly mitochondrial. It can take place in from different sources, like it can occur from lactate, from glucogenic amino acid, from propionyl coenzyme A, from glycerol. The rate limiting enzyme of the pathway is phosphoenol pyruvate carboxylase, and the regulation takes place via the induction and repression of this enzyme by insulin and glucagon, and short-term regulation can take place via covalent modification, as well as allosteric regulation can take place via activation by acetyl coenzyme A, and allosteric inhibition of the enzyme pyruvate carboxylase by fructose 2,6 bisphosphate. Or these are the important steps or points of gluconeogenesis. If you like my video, don't forget to give a thumbs up, and do share this video with your colleagues and friends. Keep watching. Thank you.